This has been a really fun meeting today. So what I want to do is return, can everyone hear me okay? Yeah. I want to return back to some of the problems that were coming across earlier in the day, which have to do with characterizing context-dependent tissue-specific enhancers and tissue-specific binding. And Matthew, of course, also touched on it just there a moment ago. So I'm sure I don't have to go into this for this audience, but it's surprising how often we see things like this presented in studies of tissue-specific binding. A lot of people still think that you can take two lists of binding sites, overlap them, and in the regions in your Venn diagram that are not within the overlap region, that's your tissue-specific binding, and in the regions in between there, you're shared. Now, of course, this approach leads to a lot of problems, and again, I'm sure no one does it here. So just to illustrate what the problems are here, I'll just show you a quick example. Here's some simulated ChIP-seq data where, it's all fake data. I've just simulated some to be differentially bound across these two conditions. So calling peaks in these two conditions separately is just setting up this arbitrary threshold. Peaks that are similarly bound across two conditions, but maybe are weaker peaks. Maybe one of them is just slightly above a threshold in one condition and slightly below a threshold in the other condition. For those peaks, you're going to get very weak specificity using a Venn diagram to call those as being, you'll get false positive differentially bound peaks. Another problem with this approach for the other end of the enrichment scale here, for peaks that have a true quantitative difference in binding, but happen to be called in both experiments. Of course, you're going to miss all of those true quantitative binding differences by just looking at your Venn diagram, okay? So we, like many others here, are interested in a more principled approach. Given that we might have multiple conditions of ChIP-seq for a given transcription factor, we want to get to this point, which is performing a principled differential account analysis using techniques that have been pioneered for RNA-seq analysis, like EDGEOR or DE-seq or other approaches like that. Now, to get there, what we're particularly concerned with is this middle step. How do we detect consistent binding events across conditions? How do we quantify the binding level at each of those consistent events? So in other words, we want to converge on some units of analysis before we go ahead with performing differential count analysis. Of course, for RNA-seq analysis, your units are genes or transcripts or something like that. There's been less attention given to how the units are defined for ChIP-seq. So the approach that we've come up with, and this is work that I presented at the RECOM conference earlier this year. It's based off of an older work that I did while I was a postdoc with David Gifford. This was the ChIP-seq analysis approach called GPS and later GEM. This approach was a mixture model approach to ChIP-seq analysis. So we modeled the ChIP-seq problem as a process that generates ChIP-seq reads by a mixture of binding events and noise. So we're going to use an expectation maximization algorithm to find the optimal configuration of binding events given the data. Another assumption in our model is that binding events generate reads according to this type of characteristic bimodal distribution, and our model is going to try to estimate the parameters or the form, the shape of this distribution as part of the learning process. And the third assumption, or the third basis of this model, is that we assume that binding events are relatively sparsely distributed throughout the genome, so our expectation maximization just has the sparse prior to reduce the number of binding events that we're using to explain the data, the observed data. So it's maybe a little easier to see what this method tries to do for those of you that aren't familiar with it, just running on some real ChIP-seq data. Here is the data, and down here are representations of the position and the relative strength of binding components in our model. So for this given region, we're starting with binding components all along the model, or all along the region, sorry. They're all equally strong. And as soon as you start running the expectation maximization process, you can see that the binding events start competing with one another to take responsibility for the reads, okay? So once the process reaches, well, Periodically, we're going to update parameters in our model, like the shape of this binding distribution, and we'll keep iterating with various runs of generalized expectation maximization. Eventually, the whole process converges. What this approach was pretty good at doing, as you can see in this example, was deconvolving relatively closely spaced binding events. So, for example, here, even though this region looks like it contains just 2 peaks, GPS was able to tell us that there were 4 distinct binding events within the region, and this was backed up by many other observations on this enhancer. So now that we want to move to multiple conditions of analysis, we want to extend our basic framework that I've presented here to integratively analyze your entire collection of ChIP-seq data for a given related collection of experiments. So the way we do that, and the way that we encourage consistent solutions, consistent binding events across all conditions, is using 2 assumptions in our model that we've added into the model. The first is that for transcription factor binding events, at least, we know that they're often associated with motifs. So we're going to incorporate motif analysis into our ChIP-seq analysis pipeline and try and encourage, using a positional prior, we're going to try and encourage binding events to align with motif instances. Secondly, we're going to have another Bayesian prior within our model that encourages binding events to align across conditions. So with these 2 forms of prior, we're going to try to encourage, but not necessarily force, binding events to align across conditions. And now, at the end of training, we're hopefully going to get a consistent set of binding events and quantification of read enrichment at each of those binding events that we can go forward with differential binding analysis on. So I'll give you another quick example of how this works. This is an example where we had ChIP-seq data in 3 different conditions. Here's the model again running on this region. So these binding components are again trying to take responsibility for the reads in this region. Now again, periodically, we're going to update parameters on our model, this time including per condition motifs. We're going to use those motifs as positional priors on where these binding events are located. And there's also this Bayesian prior encouraging things to line up. So at the end of analysis, we should have binding events that are consistently located across conditions and often associated or lined up with a motif instance. So obviously, this is most appropriate for transcription factor binding ChIP-seq analysis, not necessarily for histones or histone modifications where there's no associated motif. But I think the model itself is generalizable to other types of data once you start ignoring motif instances. So let me return to this sort of toy example here where I had defined some differential binding events, just to show you that taking the care to actually come up with consistent binding events and using this sort of probabilistic way of associating reads with binding events is worthwhile in that it gives you more sensitivity in terms of detecting weaker differential binding events than alternate approaches. And it has, like other approaches you take here, has high specificity. Let me just explain what the other approaches are. So multi-GPS, as I said, I've quantified the binding at each event using this probabilistic model. And then within multi-GPS itself, it just runs edge OR on the table of quantified binding events. So the alternate approaches would be to use some other peak finder like Max or Scissors or something like that, and to do the same thing, to quantify them, to count the reads around the peaks, or to use the peak height or something like that that comes from Max or these other approaches, and put that into edge OR. And so what I'm trying to show here is that the actual quantification that comes out of multi-GPS and the higher accuracy in terms of the positioning of those events matters, at least on the weaker differential binding. So for the rest of the talk, I want to show you how we've been using this with an example. We're also interested, like others that spoke this morning and earlier, we're also interested in transcription factor binding specificity, how a transcription factor finds its binding sites once it's introduced into a particular cell type. So from ENCODE and from many other ChIP-seq studies over the last 15 odd years, we know a lot about what what transcription factor binding sites look like within a cell type, within a given cell type where the transcription factor is active. So we know, for example, that binding sites enhancers are typically accessible. They're typically marked by particular histone modifications. They're typically bound by multiple transcription factors. What we're interested in is what those enhancers, what those binding sites look like before the transcription factor has been activated. Okay? So there's a few different models out there for what the before looks like. A number of people have shown that for given transcription factors, the sites are already accessible or already bound by other transcription factors before that transcription factor has been induced or activated. But of course, we also know about pioneer transcription factors, right, which seem to target sites on nucleosomes which are not necessarily accessible or bound by other factors beforehand, right? So we want to know how this happens for various transcription factors, what the balance of chromatin state dependent and independent binding sites is for given factors. For this type of study, what we want to do is take a few well-defined cell types and induce a given transcription factor in that cell type. And then we can compare, especially if we're doing this across multiple tissues or multiple starting points, we can compare where that factor is binding in those different cell types. And then, given a well-characterized cell type, we can look back at what those binding sites looked like before the factor was induced. So for this example, the one I'm going to talk about today is based on CDX2. For this study, we induced CDX2 in 3 physiologically relevant cell types. We induced it in embryonic stem cells, in endoderm, and in progenitor motor neurons. Now, when CDX2 becomes active in these 3 different cell types within the embryo, you get 3 different cell types being generated. So you get trophectoderm when you induce CDX2 in ES cells, you get intestinal cells from endoderm, you get caudalized motor neurons from progenitor motor neurons. So we induced CDX2 in these 3 starting cell types. We did ChIP-seq on CDX2 with a CDX2 antibody. First of all, we see a lot of locations where the binding seems to be similar across these 3 different starting cell types. For example, this HOX-C region seems to be fairly similarly bound no matter what the initial cell type. However, we also see plenty of condition-specific binding events also. And this is true even though, as far as we can see, and with all the caveats about how bad PWMs are and everything else, we don't think that CDX2 is changing its binding targets. Okay, so I'm running out of time, so I better go quicker. So we ran this data through multi-GPS, and I'm just going to show you a pairwise comparison here, but we've done this for all 3 conditions. We see plenty of sites where multi-GPS says that the binding of CDX2 is context dependent, is significantly more enriched in one cell type than another cell type. But we also see some sites where it's context independent. When we look back at, so let's take these sites for example, these sites that are bound more in ES cells than in this case, progenitor motor neurons. When we look back at what those locations look like in ES cells, we see something like you would have probably expected. These sites are already accessible in ES cells. They're already marked as enhancers. They're already bound by OCT4, SOX2, NANOG, all these other transcription factors. Conversely, again, as you would have probably expected, the sites that are bound more in progenitor motor neurons than in ES cells do not show anything special happening at them in ES cells. Okay? They're not already accessible in ES cells, and therefore maybe that's why they're not bound. So when we started this study, I assumed that these sites in here, these condition-independent sites, maybe following something like Matthew told us there a while ago, would be just open and accessible in all the cell types that we looked at. But it turns out not to be true. These sites, and in fact these sites are bound in all 3 conditions that we tested, are not accessible. They're not pre-marked as enhancers. They're not bound by other transcription factors in ES cells or in the prior state. We can quantify this a little more with SVMs. So here we train SVMs on chromatin state information that we collected from various sources. and we're just trying to train an SVM to tell the difference between sites that are bound in ES cells of these various different categories versus unbound CDX2 motifs. Sorry, okay, I'll really rush. So for the ES-specific sites, we can use this information to very accurately characterize or to very accurately predict those types of sites. The sites are bound condition specifically. As you would expect, again, the progenitor motor neuron specific sites are ES chromatin state information doesn't tell you much about them. And similarly and surprisingly for the shared sites, or the condition-independent sites. So we basically think this is what's happening at some subset of sites. CDX2 is acting as a pioneer. Basically, we're wondering what's so special about these sites. Why are these sites bound as a pioneer in inaccessible chromatin, but these sites are not bound in inaccessible chromatin? And basically, to make a long story short, we think it's sequence information. We can train an SVM using just formers to tell the difference between those sites and condition-specific sites. If we just take out the motif itself, the primary motif, just the primary motif score, the number of motif instances, and the probability of occupancy itself, those three variables do an okay job of predicting those shared sites versus condition-specific sites. So basically, we think that there are properties of condition-independent sites that are distinct from other sites. And just as a last point, I promise, we're not the only ones to have seen this for given transcription factors. There was a recent study from Rick Myers where they seemed to come to the same conclusions for ER and for GR binding as well, where they were seeing a subclass of sites that were inaccessible and, in their study, unmethylated, and which seem to have stronger motifs present at them. So this seems to be, this might be a more general behavior for mammalian transcription factors. There may be condition-specific sites that are set up opportunistically, and condition-independent sites that may be specified by some other mechanism. Multi-GPS is available, and I don't think we would have been able to come out with these nice categories for analysis if we hadn't used it. And let me just thank everyone involved in the study. This was joint work with Matt Edwards and David Gifford's group, and Esteban Mazzoni and Hinnick Richterly in New York. Thank you.
Just one quick comment. CDX2, I believe, is related to caudal in Drosophila, and we recently showed that caudal exhibits very strong self-cooperative DNA binding. So is it possible that that is what's that's what's causing, that's the discriminative feature actually, and it's consistent with more sites being present, for instance, and it's also possible that the self-cooperativity can act as to shift the equilibrium between the accessible and inaccessible states, and therefore make it.
Yeah. I hadn't seen the results, to be honest.
Okay.
But absolutely, that is consistent. I guess what I'm trying to get at here is maybe instead of categorizing transcription factors as being pioneers or non-pioneers, maybe all factors are pioneers to some extent, and whether it's by how they target nucleosomal DNA or how they have sub-cooperativity effects, we should start thinking about condition-independent binding in that way. It's just a spectrum. Thanks.
Are we breaking, or do we have to? Okay. I had one more quick question. Why? So you said you started the previous part with a sparse prior, and I wonder, and said at some point the motif matches could be used as a prior information, but motif matches are not binary. You can use the PWM to have a continuous, in which case you can have a prior which is continuous as well.
Yeah, the
is that what you were
prior is
using?
continuous. Yeah.
Okay, all right.